Hi everyone, I'm Martina. I'm in the Dr. Diego Learman's Coral Restoration Lab at the University of Miami. I'm excited to share the results of our outplanting study today that helped us hopefully predict some outplanting success. So as we all know, populations of Acropis cervicornis have been declining over the last several decades due to a variety of stressors. So it's important that restoration practitioners are strategic about where they outplant corals and factors that make corals more resilient. So there have been a variety of studies that have examined coral outplant performance throughout the Caribbean, and they've identified several factors that contribute to survivorship, including outplant density, outplanting methodologies, and phenotypic and genotypic differences of coral outplants. So at the University of Miami, we have a coral restoration lab where we have underwater nurseries, and we propagate over 40 distinct genotypes of Acropis cervicornis that have been sourced from reefs in South Florida. And so we wanted to set up a study that evaluated the different traits of these genotypes when outplanted at different reefs throughout our region in South Florida. These findings could be used to inform reef restoration efforts and help us potentially identify more resilient corals among our genotypes. So to do this, we fate tracked over 1,700 individual corals of Acropis cervicornis that were raised in our nursery. And we looked at productivity, total lipid content, thermal tolerance, which was assessed by sea bass analysis. Rich Karp presented on this earlier today, and survivorship. So we looked at these corals in our 2 nurseries and at 6 reef sites in South Florida. So we had 20 genotypes that were sourced in our nursery, and the reef sites all had existing populations of wild staghorn, and 2 of those reef sites actually had thickets. So to further illustrate the outplant study, we had our 2 underwater nurseries that had the 20 genotypes that we tracked and then outplanted at 6 different reef sites alongside wild colonies of staghorn. So we completed coral condition surveys and measured these corals 3 months, 6 months, and 12 months after outplanting. And we assessed corals for breakage or partial mortality and tried to identify the causes of mortality when possible. And any corals that experienced breakage or partial mortality were excluded from the growth analysis. But still, we were able to measure almost 1,300 corals. So for productivity analysis, we did in situ measuring of the coral individuals using a flexible ruler, and that was to calculate the total linear extension, and that was normalized to the initial outplant size and extrapolated to an annual productivity value. And then we ran a general linear model with genotype as a random effect and found that on the y-axis we have our 2 nurseries identified as KB and Bull. They had the highest productivity, which is something that we would expect to see, but we're surprised to find that the 2 thicket sites identified here as Sunny Isles and Youngs had the lowest productivity. So then we were interested in looking at genotypic variability, so we put together this heat map in R and again found that the corals were consistently fastest growing in the nurseries, indicated by the dark blue, and slowest at the 2 thicket sites. And interestingly, the genotype sourced from Miami Beach grew consistently quickly, faster, across all of the sites, whereas the corals sourced from Young's thicket grew consistently slower. And looking at lipids, we also found a significant side effect and found that lipids were consistently slower at the nursery and the 2 thicket sites. And finally, looking at outplant survivorship, we tracked this for up to a year and found that there was a significant effect of site when we ran the Cox proportional hazard model, with one of our sites, the southernmost site, being the experiencing the highest amount of mortality. And we were able to determine that a lot of the mortality was partly due to fireworm predation, but because of the amount of time between survey periods, we weren't able to identify the cause of all of the mortality. So in conclusion, we determined that outplant site had a significant impact on how corals performed, and we were able to identify a lot of the generalist genotypes among our corals, and we think that it would be best to use focus efforts on these generalist corals that appear to have high phenotypic plasticity and will do well in a variety of habitats. But we still think it's important to maximize genotypic diversity because corals that may be slow growing may have a higher thermal tolerance. And when possible, it's best to be careful about site selection and remove any corallivores or any other stressors that can affect survivorship of outplants. So with that, I want to thank everyone who contributed to this project. It takes a lot of effort to outplant 1,700 corals and track them and measure them, and we wouldn't have been able to do it without our funding sources. And if there's time, I'm happy to take any questions.
That really is a phenomenal amount of work. We do have time for questions. Anyone has a question? Yes.
That was a really nice study. I agree. A ton of work there.
Thank you.
I was just curious, did you look at any of the environmental conditions at your site, and are any of those conditions kind of predictive of the results we saw, such as with lipid content?
So we looked at temperature. We did have temperature loggers at each of the sites, and so we compared that to the sea bass analysis, which Rich presented on earlier today. But we're still sort of delving into the temperature differences. But we do think that more variability was observed at our southernmost site, which did see the most mortality. So there's definitely a relationship there.